In this manuscript, we investigated the trafficking and localization of Pten, a major tumour suppressor protein that is lost or mutated in many types of cancer. We hypothesized that Pten may be trafficked on vesicles in the cell due to two lines of evidence. The first is the punctate distribution of endogenous Pten in the cell, as shown here. The second line of evidence was that we had found Pten could interact with NDFIT1, a transmembrane protein found on vesicles in the cell. To study the trafficking of Pten, we employed a technique called biomolecular fluorescence complementation, or BIFC. The BIFC technique splits green fluorescent protein into two fragments. One fragment is attached to Pten. The other fragment is attached to a target protein, such as ubiquitin. When Pten is ubiquitinated, the two GFP fragments are brought into close proximity, allowing for the recombining of the fragments and a fluorescent signal. Using BIFC, we investigated the trafficking and localization of ubiquitinated Pten. Pten is normally observed in the cytoplasm, where it negatively regulates the PI3 kinase pathway. With BIFC, we observed Pten in a complex with NDFIT1 on Rab5 early endosomes. This complex resulted in the ubiquitination of Pten. Rab5 GTPase function was also found to be required for both the ubiquitination of Pten and the trafficking of ubiquitinated Pten into the nucleus of the cell. Separate from this pathway, we also observe ubiquitinated Pten on Rab11 containing endosomes that are known to be involved in the recycling of proteins back to the membrane surface. Our findings are interesting as they pose new questions, such as, can Pten change the phospholipid composition of endosomes, and would this result in Pten directing its own subcellular trafficking? Further research will aim to determine if Pten is merely a passenger on endosomes or an active participant in vesicular trafficking. Interestingly, our results also showed that ubiquitinated Pten can be localized to Rab11 endosomes. Rab11 endosomes have been implicated in the formation of exosomes, small vesicles that can be exported from the cell. We've previously shown that Pten can be trafficked in exosomes between cells, and this forms an extracellular communication process. Therefore, our findings here suggest a potential mechanism for this transport process. In conclusion, we've identified a new trafficking mechanism for Pten that can alter both its localization and post-translational modification. This mechanism may prove critical for the many cellular functions that Pten has in acting as a potent tumour suppressor.